Hello, everyone. My name is Maja Vukovikj, and as everyone said, I come from Uppsala University. First of all, thanks for inviting me here. It's been lovely attending the Metagenomics Workshop and also seeing some sun, since in Uppsala it's -3 degrees. I started my postdoc a year ago, and I got involved in this very exciting and challenging field of paleogenomics. And today I'm going to be talking about mapping Africa's ancient disease landscape. So let's get started. These are the topics that I will be covering today. What is ancient DNA? Where do we get ancient DNA from? What's ancient metagenomics? How to validate and authenticate pathogens as ancient? Which pathogens have been detected in Africa, and future perspectives with focus on Africa. First of all, let me introduce you to what is ancient DNA. So any form of DNA from a non-living source that shows evidence of molecular degradation classifies as ancient. It's not defined by fixed age, but rather its condition. Here I have several examples ranging from 100,000-year-old Neanderthal DNA up to Mycobacterium leprae DNA from the 20th century. And these all are ancient DNA, these all classify as ancient DNA because they have this specific, they show this specific DNA damage, and we need to employ bioinformatic tools and laboratory protocols which are tailored for working with ancient DNA. So how does aDNA degrade? So there are three processes that are involved in the degradation of ancient DNA. The first one is depurination, which leads to the formation of abasic sites. Then the cleavage of the phosphate backbones happens due to the weakening of the backbone, and this single-stranded nicks happen on both strands. With time, the hydrogen bonds between the bases break and the duplex melts, and we are left with this single-stranded overhangs. Over time, this leads also to fragmentation of the DNA. And lastly, the deamination happens on these single-stranded overhangs, in which we see transitions from cytosine to uracil. Having said that, is it possible to use DNA damage as a clock? And the answer is sort of, but not precisely, because we can use it as a clock that says today or a while ago. So the relationship between DNA damage and time is not linear, but it's heavily dependent on the temperature and the humidity from the environment. As you can see on the left-hand plot, the Scandinavian sample, which is dates from 5,000 years ago, has a lower level of C2T substitution compared to the Costa Rican sample, which is only 1,000 years old. So definitely local temperature and humidity do influence the degradation rate of ancient DNA. What we've also seen is that DNA degradation and damage varies by organism, even within the same sample. And we've noticed that between, for example, bacteria and archaea, we've seen differences in the degradation rate. And lastly, we can say that DNA damage is just a relative indicator. And on the right-hand side, this is a classical deamination plot that we usually see for ancient DNA, and this is really important for the authentication of ancient DNA, and I'm going to touch on that subject a bit later on. So what are the challenges of working with ancient DNA? We work with heavily fragmented samples, so we get a lot of short molecules between 30, 40 to 100 base pairs. The DNA is heavily damaged. It's low abundant. We usually find it, almost always we find it in a soup with modern DNA. So we have to find ways how to disentangle our ancient fragments from the modern DNA. And in order to be able to extract ancient DNA, we have to work in these clean room laboratories. Damage for us in ancient DNA is actually better than perfect, because we use the damage to authenticate individual species, metagenomic assemblies, or even individual sequences. However, it poses major challenges for taxonomic identification, accurate genome mapping, metagenomics, and metagenomics assembly. However, we've seen that the biggest challenge when working with ancient DNA is not the cytosine deamination, but rather it's the fragment length, because we can correct for the cytosine deamination either using deeper sequencing or using laboratory protocols that are specific for working with ancient DNA, and we can basically make out most of our data. However, if our fragments are really, really short, that is the limit for our studies. So as I said, the tipping point for ancient DNA studies is if the fragments are below 30 base pairs. In that case, they lack sufficient specificity for taxonomic assignment, or they just map to way too many different species, and they are biologically uninformative for us. So the theoretical limits for ancient DNA, according to a simulation study that Alan Toft and his collaborators did, is around 3 million years. So after that time limit, the DNA degrades beyond recovery for us, at least, and it's shorter than 30 base pairs. The oldest published paper is with a 1 million year old mammoth DNA, and the fragments in this study were at most around 35 base pairs. So fragment length imposes challenges also for accurate genome mapping. So our DNA sequences that we get are usually below 100 base pairs, and these lack taxonomic specificity within clades. And then again, it poses issues with phylogenies and inferring evolutionary histories. In order to work with ancient DNA, you have to have a specialized laboratory, which is a clean room laboratory. We use positive pressure, which is contrary to what in BSL-3 labs is. And the main idea is to protect the sample from us and not to introduce modern contamination into our laboratory, because we work with very, very valuable samples. Okay, where do we get ancient DNA from? So there are a few interesting sources that we can get eDNA from. One of them is seed. These are these amazing bioarchives from which you can recover quite a lot of bacteria and viruses that have been present in the person's life. And one of the sources that I'm going to talk about is the dental calculus. And this is this natural biofilm that forms on the dentine during the life of the person, and it calcifies over time. And it basically creates like this snapshot of the oral microbiome for a specific time in the person's life. And when we extract the DNA from this calculus, you can basically look at the oral microbiome. You can look at bacteria that are associated with periodontitis, or you can simply look at the bacteria that are associated with different diets. Okay, it's moving. Yeah, the second source is the dental pulp, and this is basically the material that I work with the most, and I'm focusing most on. It is made of like connective tissue, which has a conjunction of nerves and blood vessels, which through the root are connected to the circulatory system of the person. So basically, in the pulp, you can find all of the blood-borne pathogens that circulate in the person's bloodstream. And when the person dies, this tissue basically dries off, and it's kind of smeared on the pulp chamber wall. And in order to be able to extract something from the pulp, we have to drill into the pulp chamber and try to collect the mineralized tissue, hoping that we are also drilling into the pulp and collecting the pulp. Okay, so lesions are also a really great source for ancient DNA recovery. So not all diseases leave lesions on the bone. Some are very fast acting. So, but there are very few ones that are well recognized, and they do leave these distinctive lesions that you can always recognize when you see a skull or vertebra with it. On the left-hand side, you can see vertebrae from a seal hunter from Peru, which was infected with Mycobacterium tuberculosis. So Mycobacterium tuberculosis requires a high oxygen content, tissues with high oxygen content, as it is the vertebrae, and it makes these cavities in the vertebrae. And these destabilize the vertebrae, and it collapses over, and it creates this it creates kyphosis. And people with that had Mycobacterium tuberculosis usually had also a hunchback. On the middle picture, you can see a person that probably had a co-infection with Mycobacterium tuberculosis and lepra, because it has the hunchback, but it's also missing its limbs. And his lepra lives in the peripheral nerves. These people would often lose stents in their extremities and acquire a lot of different infections and lose their limbs over time. On the last, on the right, you can see a skull with a typical rhinomaxillary syndrome, in which the nasal bone has been resorbed, the maxilla has been resorbed, and also he's lost his front teeth. And this is a very distinctive look for a person with a late lepra. So definitely, these are great sources to sample if you want to find some of these pathogens. Also, I wanted to mention that other great sources are paleofeces, in which you can look at the gut microbiome of the person, blood, food vessels. You can track and culinary microbes, or sediment is also a great source for sampling and to look at for looking into ancient DNA life weight. So, what affects the possibility for pathogen detection? That is highly dependent on the sample and which bone has been sampled. So, if you're looking for a bloodborne pathogen, the pulp is the best source that you can look into for a specific pathogen. If you're looking, for example, for parvoB19, you would probably want to sample some long bone, because it is most likely to be detected in the bone marrow. The sample preservation plays obviously a very big role in the possibility for pathogen detection. The depth of sequencing is also very important, and of course, we can sequence very, very deep, but also it highly depends on the library complexity and what we have in our sample. So if we have a poor library complexity, regardless of how deep we go, we will not be able to recover more of what's already in. The tissue tropism, you have to get to know your pathogen a bit and to know if it preferentially or exclusively replicates in a certain tissue. Then the abundance of the organism, perimortem, is really important. The higher, of course, the better. So when looking for bloodborne pathogens, we hope that the pathogen that we're looking for was actually the cause of the death of that person, so that we would, in that way, we would have the highest abundance of this pathogen. And the genome size and composition, whether we are working with single-stranded or double-stranded organism, highly affects the possibility for detection. What is ancient genomics? That is the next thing that I wanted to talk about. So by definition, metagenomics is the study of the collection of genes and genomes of the microbiota within a given environment. The same definition applies for ancient metagenomics, but in addition to the antemortem host and microbial genetic material, ancient metagenomes almost always contain at least some postmortem DNA from the thanatobiome or the necrobiome. And to get things even more messy, they are from various ages. So we get bacteria that have been involved in the degradation process of the body in the initial stages. So those bacteria die off, and they acquire the same deamination profile as our pathogens of interest. So that makes the analysis really tricky, and yes, we need to pay a lot of attention not to make some wrong assumptions. So the most accurate metaphor I could think of for ancient metagenomics is a puzzle. So the metagenome looks like a collection of 100,000 different puzzles. And then in the microbiome, we have these different bacteria and viruses present. Then we have several hundred different puzzles that belong to the necrobiome. And of course, we have the endogenous DNA, which is the human DNA. When we get the metagenomic data, it looks mostly like this. So we get many, many different pieces of the puzzle, and we don't know which one goes where and to which organism it actually belongs. And the most basic question at the beginning that we have to ask ourselves is who is there and how to get from raw DNA sequence to taxon tables. And the only way we can do that is by using taxonomic profilers. So there are several different options, alignment-based and alignment-free, and I'm just going to briefly talk about Wald and Kraken, which are the ones that I mostly use for my analysis. So Wald performs taxonomic assignment by aligning to a reference database. You can use different database. You can either do RefSec or NCBI NT, depending on the cluster that you're working on. It's quite slow and memory intensive, but it maximizes the data available. The database is customizable, so you can also build project-specific databases, and uses an LCA, which is like lowest common ancestor algorithm, which is really helpful for ancient DNA. And the downside is that it's very computationally intensive with large databases. So in order to fit 40,000 species database on your cluster from Malt, you would require at least 1 terabyte RAM. So definitely it's very computationally hungry, and you have to make sure that you have enough resources to be able to run this program. Yes, so I just wanted to show you this sketch and to show you that from all of the reads that we get from a sequencing run, very few of the maps we do get mapped, because that's mostly due to the short reads, the database bias, or the database composition. Kraken Unique is a taxonomic profiler that works by k-mer matching, and it's much faster and less computationally intensive compared to MALT. The database is also customizable and uses LCI algorithm. It can be prone to false positives, and it doesn't provide alignment data. So you would have to, after using Kraken, align the data and build your deamination profiles to verify that you're working with ancient DNA. So in the past few years, there have been a lot of publications about comparison between different taxonomic tools, and the take-home message would be that there is no such thing as the best taxonomic profiler. You should just use what's best for your project or your research question. These give broadly the same results, all of these taxonomic profilers, but you also, yes, as I said, you have to think about your research question and what would fit best your study. And if you get rid of these low abundance reads or low abundance taxa, then you would most likely get very low number of false positive detections. In the performance of the taxonomic classification, a huge importance have the database that have been used. These are really important, and it depends on how big the database is, how inclusive it is, how many species it has, and also another thing that you need to think about databases is that sometimes these are contaminated, and when you use some of these, you might just get some very weird results. So you have to be careful about the databases that you're using. Yes, and another sketch. Just I want to show you that almost when we do taxonomic classification, almost around 85 to 90% of our reads are unclassified, and we get a very high, low number of reads that actually does get classified. So the question is, what do we have there in all of those unclassified reads? And yeah, that's an interesting. It's an interesting thing that I think that needs to be further looked into, and maybe there are some interesting new viruses or bacteria in those unclassified reads. So how to validate and authenticate ancient DNA? So the results from the classifier should not be the end results. For ancient DNA, the taxon validation will often consist of 4 steps, which is authentication of the data as ancient DNA, evenness of coverage and edit distances, competitive mapping to relevant reference sequences, and identification of intervals specific to the target species or subspecies. As I showed previously, we see these are so-called smiley plots, and these show the deamination of the DNA. And if you don't see any deamination pattern for your sample, you're most likely working with a sample that's not ancient. This is an almost perfect deamination plot that you get for ancient DNA. And what we usually do get for pathogens is something between this perfect smiley plot and this very spiky one. And you get very spiky plots if you work with reads that are below 1,000, or if you have not filtered properly your reads and you've left some reads that are around 30 or below 30 base pairs, so they introduce noise in your data. And also, it's really important to know what's the cutoff for the C2T transition frequency, and what qualifies as ancient in your sample. And then lastly, we have this final plot where you see that the sample, the reads have been mapped to a reference, but it's most likely due to the elevated baseline, it's not the right, but it's not the right reference, but very close, a close relative to the organism of interest. Another thing that we need to look into in order to be able to authenticate ancient DNA is its fragment length. So you have to see fragments that range between 30 to 100 base pairs. If you're seeing longer fragments around 150 base pairs, then you're most likely looking at modern sequences or contamination. Okay. Here I'm giving a few examples of what a false positive finding looks like. So this is on the upper side. You see an IgV snapshot from a false positive finding. You see all of the reads have clustered in only a few positions on the genome with very high depth. So if you just look at your mapping statistics, or you look at the number of taxonomically classified reads from Kraken, and if you see a high number, it might lead you to make a wrong assumption that your organism of interest is actually present. But if you look at the evenness of coverage, and if you plot it, then if all of your reads cluster on one or a few positions in your genome, then definitely that's not a positive detection. But if you see a nice breadth of coverage and with similar depth, then definitely you're looking at a positive detection. And yes, here are also two examples of positive and of true positive and negative detection. As you see, all of the reads are distributed nicely along the reference genome, whereas for the negative detections, you see some spikes on the reference with where the reads have mapped in high depth. But that is most likely due to some conserved genes in that regions that map within the genus or the family of the organism. Also, edit distances are quite important to look into. Edit distances are actually the number of mismatches between the reference and the fragment of interest. So if you have 2 to 3 mismatches, then you are most likely looking at an ancient genome, because depending on when it was sampled and how far back it goes, you should have some divergence from the modern reference. But if you are looking at 5 or 10 mismatches, then maybe you're aligning at the wrong reference, or if you're lucky, you would find something very, very divergent, but you would have to do a lot of analysis to confirm that. And another thing that you need to look into to confirm that the presence of your pathogen in your data is to identify in the specific intervals to the target species or subspecies. So, as I said, you have to get to know your organism of interest, whether it has some accessory DNA, such as plasmids. I'll give you one example about Bacillus anthracis. This is a bacteria that I found on my first run, and I was really happy that I've actually found anthrax. But that's actually not the case, because this anthrax, Bacillus anthracis, is very common in soil as well. So when I aligned all of my reads, I saw that none of my reads mapped to any of its plasmids. It has 2 plasmids, and it turned out that this is just simple soil bacteria. So no big paper for me there. Then also Yersinia pestis has three plasmids that you have to have maps reads aligned to it, and as well to the bacterial chromosome, to be able to convince your reviewers that actually you found pestis in your sample. And also specific genes or gene intervals, such as the YMT gene on the PMT1 plasmid in Yersinia pestis. Yeah, you have to, some species have specific intervals that you have to have reads aligned to in order to be able to publish. So what are the metrics that point to a positive hit? I'm just going to summarize. You have to look at the breadth and depth of coverage, deamination and fragment length, presence and absence of plasmids, if there are some, presence, absence of specific regions. Then do competitive mappings, basically force your read to choose between several different references. And lastly, you have to think logically whether it actually makes sense to detect this organism in the sample tissue. So, which ancient pathogens have been found so far? In Europe, there have been a lot of different pathogens that have been recovered so far. So Yersinia pestis was found in several different sites. Then Mycobacterium leprae, Mycobacterium tuberculosis, HBV as well. These findings, these findings have, or similar findings have been found for Asia and for the Americas. However, no, we don't have any systematic findings or papers for sub-Saharan Africa. For example, Mycobacterium tuberculosis emerged most likely 6,000 years ago, and which is contrary to what was estimated to 70,000 years ago. But that phylogeny was built based only on modern genomes. So when you are able to pick up modern genomes, you can basically refine the phylogeny, and you can estimate the most common, most recent common ancestor for the pathogen of interest. If we didn't have ancient DNA, we wouldn't know that the same bacteria was responsible for the Yersinia plague and Black Death. And also there had been findings of Yersinia pestis that dates 5,000 years ago. And in those lineages, Yersinia pestis does not have the YMT gene, which is the murine toxin, which helps for the colonization of the gut of the flea. So with time and over its evolution, it has acquired like key virulence factors, which has enabled the bacteria to cause the bubonic plague. For hepatitis B virus, there have been a lot of findings in Europe and in Asia. So in this big paper by Kosher and his collaborators, they've managed to recover genomes of around 140 HBV viruses, and ranging from 11,000 to 400 years old. And basically, in that data, we can see that there have been two major lineage turnovers over that for HBV over that period of time. There is this ancient lineage that circulated up until from 10,000 years to around 8,000 years ago, and then we have the emergence of this Bemba lineage, which is which took over and was prevalent in Europe for around 4,000 years ago, and then that lineage completely disappeared, and the modern HBV viruses started circulating and diversifying. Not going. Okay. So, as I said, no systematic pathogen survey exists for sub-Saharan context, and all of the ancient DNA studies basically focus only on Egyptian mummies. And one of the key studies that has been published for Egyptian mummies is the study by Nukam and his collaborators, in which they managed to recover Mycobacterium leprous strain from 2,200 years old, and hepatitis B virus, which dates to 2,000 years ago. And this is the oldest Mycobacterium leprae that has been recovered so far, and it kind of anchors the phylogeny for Mycobacterium leprae back to its most recent common ancestor, dating around 6,000 years ago. And then hepatitis B virus from this study clusters really nicely with modern African and Brazilian strains. For the other studies that have been published from Egyptian mummies are only PCR studies, so they have not recovered the full genomes, and there hasn't been no phylogeny. They've just shown that these protozoans existed and circulated in Africa for many years. This is the only study, metagenomic study, that's been published for sub-Saharan Africa, and that's the study of the Balito Bay boy. It's been this is a study published from our group, and we managed to recover Rickettsia felis, which is a flea-borne pathogen, and it causes typhus-like rickettsiosis. And the important thing about this is that I think in 1994 was the first study published about Rickettsia felis, saying that this is an emergent pathogen. By finding this in sub-Saharan Africa, in a 2,000-year-old boy, we actually show that this is a pathogen that had been circulating here in Africa amongst hunter-gatherers who did not practice any animal husbandry for almost 2,000 years ago, and it is definitely not an emerging pathogen. So this is the thing that probably most of the people here are interested about, and that's what have we found so far. So I just wanted to show here on the map all of the collaborators, collaborating countries basically, and all of the sampling locations. So we have samples from the DRC, from Zambia, Zimbabwe, and South Africa, and our samples range from 10,000 years old to 200 years old samples. So we've recovered double-stranded and single-stranded DNA viruses, bacteria for which we know that have high prevalence in Africa now, and bacteria that have caused major epidemics in Europe in the past. However, since we're working with highly fragmented and damaged material, we had to build an Africa tailored capture panel in order to be able to enrich our samples for the viruses and bacteria of interest, and to be able to recover their full genomes, and then to be able to do phylogenies and look into their evolutionary histories. And then RNA viruses are also a very interesting subject that I don't have time to go more into detail, but we are working also on an RNA project, we will just explore the technical limitations for RNA recovery. Although I can say that that it is possible to recover RNA from ancient samples, and it has been done so far. And I'm just gonna stop here. Okay. And the future perspectives for pathogen discoveries in Africa. while going through papers, I found some interesting things. There is like a written document that dates a while back, saying that actually the Justinian plague originated from Ethiopia, and that it happened during the fifth or sixth century, because the Ethiopians had really close relationships with the Byzantine Empire over the Persian War, and in that written record, it said that most likely the plague entered Europe through the ties between the Ethiopians and the Byzantine Empire. So this is a really interesting thing that we can investigate if we do manage to find something. Yersinia pestis, so it has a lot of active foci in Central and East Africa, but Madagascar stands out by the number of plague cases, and the reason for that is that it has incredibly efficient reservoir vector combo. So it has the black rat and also the Asian shrew rat. That make this perfect reservoir that always gets infected by these Xenopsila brasilensis and Xenopsila chopis fleas, and then the island ecology, because there are not many predators there. All of the reservoirs and vectors are always present in high abundance, so the bacteria can. Sorry. So yeah, the bacteria can be spread. The people in Madagascar usually keep their grain into their houses, which allows for close contact between the humans and the rats, and the seasonal highlands climate, which is cooler and drier, also helps for the maintenance of this, for the reservoir and the combo. So in Madagascar, Yersinia pestis arrived through an Indian trade ship in 1898, I think, and when it was introduced over the third pandemic, it then kept diversifying in Madagascar, and it still has active foci even today. So it would be interesting to look back in the history and in these first strains that came to Madagascar and how it diversified over time. So we've already picked up Rickettsia felis, so we want to speak broadly for flea-borne rickettsiosis, because we also know that Rickettsia felis nowadays has a high prevalence, around 50% prevalence in Africa, and it also can be found in asymptomatic patients. So it would be interesting to see how big of a prevalence this bacteria had in the past. Then Salmonella enterica typhi, there are written records, and Africa has a long track record for outbreaks with Salmonella typhi, but there are no identification made so far, and because this can be found in the blood by sampling the pulp, we hope that maybe we can find some traces of this bacteria that had been infecting people in the past in sub-Saharan Africa. So the Mycobacterium tuberculosis ancestor is inferred to have originated here in Africa. So we really hope that we might be able to pick up something and prove that indeed its ancestor strains did emerge here in Africa. But yeah, you would have to wait and see what our results will show. And also Leishmania donovani and Trypanosoma brucei, they have high prevalence in Africa. The Trypanosoma now is not that prevalent, but most likely they had high prevalence in the past. So we hope to be able to find this in our ancient samples. And Plasmodium falciparum, which accounts for 95% of all malaria cases in the African region. So Africa as a key region for past and present epidemics provides unique potential for understanding the global infectious disease dynamics. And the ancient genomes allow us for direct comparison of ancestral and modern sequences. And by having them, we can have insight into the virulence evolution, transmission, and refined molecular clock calibrations. So we can have a look at the deep time calibrations of the pathogen divergence rate, reconstruct the virulence trajectories, or refine models of emergence and spread. Having said all of that, Africa remains profoundly understudied when it comes to ancient pathogens, and we hope to help to close that gap. Thank you very much for listening.
Hello? Oh, there we go. Wow, that was really, really interesting. One thing that I would like to ask is, obviously, how likely, if you find bones, is it that you're going to get anything meaningful out? And then you chatted about temperature and humidity. So obviously, what you get in the Siberian tundra is probably perfect, whereas here in Africa, it's a bit of a problem, especially in the tropics. So yeah, how often do you find something in African ancient DNA? And
yeah, so our group has a relatively good track record for extracting ancient DNA. So I'm not sure about the number, but I think that we have like 20% success rate in recovering ancient DNA from samples. So yeah, of course, it depends from where these samples are coming and how porous the bone itself is. So that is absolutely a limiting factor for us. But as I said, it is possible, and yeah, we've shown that it can be done. And as you said, there is a difference between the DNA and the RNA that might be extracted from the Siberian tundra compared to our samples, and definitely we see highly fragmented genomes, and a lot of deamination on the sequences terminium. But we do manage to pick up signals that the pathogen was there. But in order to be able to prove and to build phylogenies or look more in details of the virulence of the pathogens, we definitely need a capture panel to do that.
Yes, Alex.
Thank you very much for the talk. Just to know, in fact, we know that now for most of bacteria, we have antibiotics, we have vaccine, and all those things. How these ancient genomics or metagenomics can help to control disease now? Because we know that maybe genome have changed with time, maybe bacteria have adapted with the new context. How all those investigations can help to control disease now? And also my second question is concerning zoonotic infection. I don't know if you also try to work on maybe animals, also to understand the link with humans, something like that. Thank you.
For the animals, that is that is something that we would want to do, and we would want to look into animal bones and to see whether and which pathogens we can recover, and whether there have been zoonotic spillovers in the past. So that is definitely one of the projects that we would want to work with. For your second, for first question that you talked about, that you asked me. So basically, if you want to look at AMR genes, what we can do with the ancient DNA is we can track when these genes were acquired, let's say, because they're most likely to be found on the plasmids of the bacteria. So we can track when these bacteria have acquired and how these genes have evolved through time. And also the information that we have about ancient pathogens will help us refine the phylogeny, will help us determine the substitution rates for the pathogens, and refine like disease models or make predictions for future pandemics.
Okay, I can just talk. So thank you very much for the talk. My questions are kind of random and mostly just out of interest. The one question is, we touched on it briefly in the workshop today, but you were speaking about Ethiopia and Justinian times, the Byzantine, and it sounds like a relatively politically loaded landscape if you're going to sort of attribute, beginnings of epidemics or pandemics, or the source, the root place of any kind of pathogen. I just wondered if that is something, and whether there's difficulty in navigating that space with reporting findings. That's the one thing. The other thing is obviously looking to the future, given that there's so much that hinges on how specimens are preserved now for your analysis. Is there a way when we die or animals die that we should be preserving their tissues for future generations to better understand?
That would be great for future research. If people in 1,000 years from now can get better insight in the viruses and bacteria that we've been infected with. I don't. Yeah, we should think about it. So I think it's a natural process. So I think that in the future, probably even the methods for analysis and the extraction procedures would be even better. So they would be, people from the future be able to even better recover the bacteria or the pathogens that we've been affected with. And for the difficulty of publishing our findings, I don't think that I haven't heard that any group has had problems with publishing papers for the origin of some bacteria or species in a region, a specific region. In the past, people have been mostly skeptical about whether you're looking at an ancient organism or a modern organism. So it's in 2012, when the first Neanderthal genome was published, there was a lot of skepticism whether that indeed is an ancient human. And the field has advanced quite a lot, and a lot of tools have been developed to basically to authenticate these sequences as ancient. And I think that was mostly the limiting factor for publishing, proving that what you're seeing is actually ancient so far.
Thank you very much for that very interesting talk. I have two questions, and the first one is looking at this kind of work, and also from your experience, are there any ethical concerns and issues that you have had to navigate to get to the samples, or at your institution level, even in some of these countries? And two, looking at how diverse the African population is, even genomic-wise, do you think such work can move forward maybe to describe some of the African ancestry? Because if you go like to Uganda, Rwanda, the Bantus, and you come to South Africa, there is that similarity. So do you think this kind of work in the future can answer such questions of African real ancestry and lineage?
Thank you. So we don't have to get any special ethical permission from ethic board to be able to carry out such studies. However, we need to get consent from heritage councils from the country from which the samples originate. So that's the only permission that we need to basically to get. And also my group, actually, we are producing these metagenomic libraries, and I work only on the pathogens. And my group is mostly working with the human DNA, and yeah, they're tracking African ancestry, and they work with very different African populations here. And yeah, I can show you our group's page, and then you can, if you're interested, you can go through the papers and what has been found so far.
Okay, fantastic talk from Maja, and we are going to have to cut. I'm sure there's a lot more questions, but we'll have to answer those after the end of the talk. Because now we have a talk from Erik Karlsson. So while he gets set up there, I'll do a bit of an introduction. So
Good.
you good? Okay. You feel like there's a problem. So Erik is here from Cambodia. He is the director, or the head of the virology unit at Institut Pasteur in Cambodia, as well as the director of the National Influenza Center of Cambodia, and also head of a couple of different reference labs as well, the H5 and COVID-19 reference labs. But his interest is really in developing early detection surveillance systems for emerging pandemic potential viral pathogens. And he's going to talk to us today about some of his cutting edge techniques for detecting emerging pathogens.
Thanks, Erik. So thanks for sticking around, though I know most of you kind of have to because you're part of the course. And I know that I'm the only thing probably standing between you and a glass of wine and some food. So I'll try to keep it engaging, at least to keep you awake so that you're ready for your wine a little bit. So I'm gonna talk about more modern things. Very, very nice talk from Maja about ancient DNA and how we measure that. I'm going to talk more about a little bit about how we look at high-risk interfaces. So we're going from things like bones and humans to places where animals and humans in the environment interact. Okay? You're right, this thing doesn't work unless you point it that way. So just a little bit of background on my unit at Institut Pasteur in Cambodia. We have been at the animal-human interface for over 29 years. We have an extensive experience with doing studies that are in a One Health context where we're looking at spillover from animals to humans, even before it was called One Health. And so we have very extensive biobanks and very extensive experience looking at these kinds of interfaces. And some of the things that we look at, again, I've just listed some of them here. So you can pick your favorite virus out of this list if you want, but you can see that we do study influenza and a lot of different things. So most of the stories I'm going to tell you today are about influenza, really. Plus, it's my favorite virus. So what we really try to do in my laboratory is do early warning. So I'm very interested in early warning. So basically, how do we anticipate what diseases are potentially going to emerge and understand the risks? How do we prevent those from actually spilling over or reduce them? But to do that, we actually need to detect what is going on, right? And then, of course, we need to be able to respond to them, either when they're spilling over or before they spill over. So these are really what I try to focus on in terms of my whole vision for my unit. Of course, we do a lot of other public health and animal health and things like that, but overall, we're really trying to look at these high-risk interfaces. So something like a live animal market, a bird market in Asia, but these really require a lot of active surveillance. And anyone that's done active surveillance in places like bird markets or anything like that know that this is really, really costly and time consuming. Okay, it takes a lot of people, it takes a lot of effort, it takes a lot of being out in the sun and things like that. So, it's not easy. And it's often not done in the easiest of places. So, we're going out on lakes, we're going into bat caves, we're going to many other places. And you can see Jay, I think right there, that was you, right? Yeah. So, these people are going out and actually, having to get samples from very, very remote places. Environmental samples. So compared to your traditional sampling options where you're capturing animals or you're going to swab people or doing things that are quite difficult, environmental samples really present a viable option for early warning because we can use them for things like in-field diagnostics and really getting a good idea of what's happening in that area very, very quickly without having to do a lot of sampling and a lot of dangerous things. So they not only increase our speed of collection, so you can imagine it takes much less time to go collect some swabs from surfaces and some air and some water and things like that, than capture 100 birds. They're comprehensive. Basically, you're looking at that entire environment as a snapshot. Sorry, this microphone is rather funny. They improve biosafety, right? So when you're not handling animals that potentially can pee, poop, bite you, do things like that, it's much, much better. I'm switching microphones. What they also do, and what people a lot of times in public health don't think about, is they increase animal welfare. Anytime we're handling animals and things like that, we don't want to stress them out. We don't want to go to a farm and kill all the farmers' animals from picking them up and sticking swabs in places they probably shouldn't be, right? And then they reduce cost. Everything's about cost, right? So if we can reduce cost of what we're doing, we can do more of it. And we can do it faster, we can do it better. So what can we sample? Most people, when they think about environmental sampling these days, it's wastewater. Wastewater, 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 wastewater. And that's great because wastewater was used quite a bit during COVID-19, but wastewater's been around since the 1970s with polio. You can even argue that it goes back to John Snow and the pumps, in London, where we're really looking at waterborne infection, right? And it's a really great example of environmental surveillance, but there's a lot of other options for us to look at. So, there's water, but there's also things like soil, surfaces, feces and urine. Some people would argue these are direct animal samples, but we can call them environmental as well. Then things like other people are looking at things like feathers for birds. I've seen people take spider webs. I've seen people take dust, but also things like air in places that we potentially are looking for respiratory pathogens. So this is a live bird market in Phnom Penh. This is the largest live bird market in Phnom Penh. This is just one of the slaughtering stalls. So sorry if this is right before the end of the day, but you can see a lot of things going on here, which I'm sure that many of you could point out as risky behaviors for zoonotic spillover. Yeah. So I won't have to go through that. But there's a lot of different places that we can actually look for virus here. So there's things like feathers, there's waste and wash water, there's cages, there's surfaces, but there's also things like air. And that's really kind of what I'm going to talk about today and what we've been focused on for the past couple of years, because we've been taking this kind of sample for 10 plus years. Okay. This kind of sampling we really started during the COVID pandemic. And so we've been doing this in a lot of different places and with a lot of different samplers. So these are some larger samplers that we've been using, these thermoscientific ones. They're quite nice. Again, this is in the holding area of that same market. You can see lots and lots of birds here, a sampler. This is in a slaughter area of a different market in Cambodia. Again, this is a slightly different slaughtering setup. It's more outside, things like that. But we've also been using different kinds of samplers. We have smaller ones now. You might recognize this guy right here. We have smaller ones now that are actually able to go in more places. They're easy to carry around. They're battery operated. So the technology keeps improving, so we get to keep playing with more and more toys, as it were. Oop. Skipping around. We also do this in other places. So for people that have never seen this, this is a bat farm. So it's a guano farm. What's actually in these little palm fronds up here are these small little Scotophilus bats right here. There's about 30 to 60 of them that live in each little one of these palm fronds. They live there quite happily. They fly out at night, they eat insects, and then they poop on the ground, and people pick up all this poop for guano for fertilizer, okay? And these farms are all over Cambodia. And so we've been doing a lot of work here in things like bat urine and feces and soil, but we've also started doing things like air sampling underneath these guano farms. And so here you can see our teams collecting samples in these farms with air samples as well. We also do things like fruit bats. This is a fruit bat roost. So this tree right here is actually in the middle of Phnom Penh, right in the middle of the city. And we're actually about 5 minutes away from our institute. You can see people in the background sampling, and we're taking different kinds of air samples near the roosts. This is actually, we're also doing this right now in Australia with our collaborators. We take these things into caves. So here you see the team in a cave, air sampler. Remember, they have to lug these 20-kilogram air samplers into the caves and things like that. There's a lot of other surfaces we can take in these caves. And then they can do things like put them outside while the bats are flying out and really get an idea of what's going on in that entire colony. And so this has happened several times where we've placed samplers here. There's about 1 million bats flying out of a cave in Battambang, and you just can run it while those bats are flying out. We can do this in other ways in these caves. This is that same cave that you saw in the other slide where the bats are flying out. But of course, we want to look inside the cave as well. So we can take these new samplers and bring them down into the cave. This is actually one of the workers that actually farms guano in the cave. We had to make him put on PPE to actually take this video. But normally he would go in just a loincloth and baby powder, if I'm correct. So he was not happy to be wearing this PPE, but for ethical purposes, we have to do this. But you can see we can get a lot different kinds of samples down in the middle of the cave versus outside on the ledges. We're also doing it in places like swine abattoirs. Of course, these are also high-risk interfaces. These are all different pigs from different farms. They're spray painted to indicate what farm they've actually come from. And we've been doing swab feces and surfaces, but of course, air in those places as well. And then we can use other kinds of samplers, things like these handheld samplers. So this is a small biochemical sampler that was originally made for outbreaks in mink farms in Denmark. And these are really good for very high pathogen loads. They're not the greatest for understanding risk, but they're really great to get into small places. So, you can really target what you're looking at. Ignore my hand without a glove on right there. But we do actually run these side by side with our other samplers, just so that we have these samples to be able to take them and collect them. Then we can also do things like if we see pigs coughing, we can actually target those pigs as well. People are now doing other things. I don't know if you guys know Andy Bowman from the US, but he's working with swine farms and things like that to do acoustic monitoring of pig farms. So whenever you hear a pig cough, they actually have air samplers that actually turn on within the farm that are triggered by pigs coughing. Okay, so you can use these in a lot of different ways to actually monitor the farms and things like that. We can also use them to get close to wild animals. And this is a civet, if anyone's never seen a civet before. They're swabbing it while also trying to get air samples out of it just to see if it works. We've used this successfully for FMD outbreaks in elephants and sun bears and things like that, at zoos and things like that. So it works for a lot of different kinds of pathogens. And we've even done things like try to strap them to drones and fly them around because they're quite light, or to what I call terrestrial drones, which are really just $60 cars that we buy at the grocery store. People have been flying these in caves. Again, you'll see Jay here. And then we can do things like drive them around farms and things like that, which is really, really great because if you go sample, if you've ever gone sampling on farms, you know that you have to then leave the farm, take a shower, potentially stay another night, and then go to another farm. Here, you can potentially imagine having 5 or 6 or 10 cars that you go into 5 or 6 or 10 different farms that day, collect all the samples, and then bring them out again. How else can we utilize this technology? We do these things in a lot of different places. My team goes out and teams from IPC go out for a lot of different reasons. And we can use the drones and other technology to do things like look at wild bird colonies and actually count how many birds have died or how extensive that is spread. So this is actually a bird colony in the middle of a lake in Cambodia that's been affected by avian influenza, and you can see a number of dead birds there. We can use the handheld samplers for actually going to handle those dead and dying animals. And we can use them for functional surveillance by looking around this colony and finding things like these semi-free-ranging duck farms that may actually be facilitating the spread of these viruses, because these farms actually move over the course of the year, because it's cheaper to actually move a duck flock around and let it feed on rice paddies than it is to buy food for them. So they'll actually pick up these entire farms, put them in a truck, and move them to the other side of the country. You can imagine them taking those viruses back and forth over time. So a big question I get all the time is, does this work? And so just on a basic PCR level, so using a gold standard, you can see very clearly if we're looking at air samplers from slaughter, holding, and then this is actually a food sales area, which is completely physically separated. You can see a very clear pattern of detection where obviously we're seeing the most viral loads in the individual poultry, but then you can see it stepwise from slaughter to even this food sale outside is able to detect avian influenza in the air. We've also done studies in the past where we've made people wear these personal air samplers. These were originally made for construction, and they're set to breathing rate. And so there's this tube here that's extending up and actually coming out right by his mouth. And so when they wear these, and we actually can culture out of these air samplers, there's 100% chance of us actually catching a viable H5 or H9 virus within 30 minutes in these markets during peak season, okay? So these people that are working in these markets very closely with these birds and things like that are being constantly exposed to these viruses. And as soon as you walk into them, you're probably exposed as well. And one of the things that we're trying to understand now is why these people don't actually get sick. How else can we improve surveillance for early warning? So I've talked about sampling, but what about testing, which is, we'll get to the metagenomics later, but there's also some other things that we can do. We want to do a lot of things to improve what we're doing, not only in the lab, but also in the field, obviously to increase and ensure sensitivity and specificity, but also increase the speed and versatility of the diagnostics that we use. And then, of course, discover new pathogens or high-risk areas with pathogens of known risk. One thing that has been very, very critical for us lately is really integrated indexing and really speeding up our nanopore processes. Any minutes or seconds or hours we can shave off of the ability to get results from a clinical sample or from a bird or anything like that is absolutely critical to response. And so we've done a lot of work with our collaborators at Johns Hopkins. This is now a commercially available kit from NEB. Actually, it was just released last week. And basically we've integrated the index for nanopore barcodes into the whole genome sequence primers for avian and for influenza, which are universal. And so basically you go from, what people were doing was 33 to 72 hours worth of sequencing. We can now do that in about 7. So even from this weekend when we had our last H5 case, our sample was there at 8 o'clock in the morning, and it was on the sequencer at 5 o'clock, and we were already getting data. Okay. We've coupled this as well, and we're still working on this with field forward real-time bioinformatics. So we actually have a program now that allows us to instantaneously be able to read these samples in real time after 10, 20 minutes they've been on the sequencer and quickly understand what we're seeing in those samples as well. This has been critical for us for a number of different subtypes, and I won't go too far into influenza subtypes today, but things like H7, H9, and especially H5, but also discovering new understudied subtypes of influenza in poultry or things that have never been detected in poultry before. And then, of course, responding to cases of human spillover in Cambodia. Not only was that, H9 cases in 2021 and 2022, but of course, the H5 cases that we've been having very recently in Cambodia since 2023. And the sequencing has really been able to allow us to link the poultry and human cases together so that we understand that these are all cases of natural spillover. There's no human-to-human transmission. But what we've also seen is that in the course of this infections, all of these new infections, except for 2 that happened very early, these have all been caused by a reassortant genotype. So we've seen some really crazy evolution of the flu of H5 within the greater Mekong subregion. And we think this may be associated with why we're seeing so many more cases in Cambodia, including the 18 that we've seen this year, the most recent one being last weekend. It's really quite a critical situation, and sequencing in genomics and rapid sequencing in genomics is absolutely critical. And some people may argue with me, oh, it's not sensitive enough, or, we get a lot of errors with nanopore. The new nanopore chemistry is quite good. We've proven that. And for me, speed is sometimes more important than depth of sequencing or anything like that, because I need to know, I need to inform the government of what's going on. And you've seen that with Ebola, you've seen it with COVID-19, things like that. And we utilize that quite a bit in my laboratory. Not that I'm saying that Illumina and other sequencing is bad in any way. Let me just keep an eye on the time, just so I don't keep you too far from wine. Another thing that we've been doing is multiplexing pan-viral family assays. So if anyone remembers back to PREDICT, was anyone involved in PREDICT? PREDICT 1 or PREDICT 2? No? Okay, maybe that dates myself a little bit on this one. But PREDICT 1 and PREDICT 2 were really the first big One Health kind of studies. They were using pan-viral hemi-nested PCR, which if anyone's ever done pan-viral family hemi-nested PCR, it takes a long time, it's dangerous, it's going to contaminate your laboratory. But it's really useful, right? Because you can discover coronaviruses or orthomyxoviruses and things like that in one shot. So what we did was we took these and tried to say, can we couple environmental surveillance with these pan-viral assays to really, move forward with field-forward diagnostics and be able to do things like test things in caves, right? And so what we did was we integrated the nanopore barcodes into these pan-viral family assays, but they've actually been changed into single round PCRs now. So everything is the same amplification temperature, same kinds of replication. So we basically have a toolbox of different kinds of viral families. And I can say, okay, today I'm studying bats, or today I'm studying birds. I want orthomyxoviruses, I want filoviruses, I want lyssavirus, and I want corona. Mix them all into one pot with one barcode, and we can immediately run those on a PCR machine. And we've been doing this in places that are rather remote. Here you can see the setup. And really, it's a very minimal lab setup. And this is my postdoc, Anna, who's running the samples now. And so we've been running them on these bento boxes, which are just basically toy PCR machines that have been made for remote work. And so she's done a very, very, very interesting job looking at how we can use these on numbers of different sample types. But also at this point, she's integrated speciation into everything that we're doing as well. So now we can actually run the multiplex pan-viral assays as well as the species. And she's been able to do things like if she takes a tick blood-fed tick, she can determine the species of the tick, the species of the dog that are fed on, and whatever viruses are in both animals in one sample. Okay. You can see here, this is just an example of things that she's doing. So we're looking in things like wash water, air, birds, humans, bats, dogs, and things like that, and detecting all these different kind of viruses. And we've done artificial experiments where we've literally mixed all of these viruses together in one tube, and we're able to detect all of them in one tube at the same time. So you can potentially get a quick metagenome as long as you're looking for certain viral families. And we can do it anywhere. So this is all done on a nanopore machine. And again, we have real-time bioinformatics to do that. Apologies, I don't have a video of that running on this, but I can show you later. Finally, and sort of the whole point of this talk, I think, is, can we take these environmental samples and combine them with metagenomics for even more information, okay? So if we're going to go take these environmental samples, and we're going to bring them back to the lab for now, can we get some really, really interesting information out of them if we do some deep kind of analyses? So I don't think for this crowd, I necessarily need to go through why metagenomics increases the utility of environmental samples, but just to remind ourselves that metagenomics is really useful in many different ways. Not only is it unbiased and can discover things, but we can do a lot of things like understand function of those viruses and look at other things that we're very interested in, known viruses. So I just wanted to walk through a few questions that we had, and we get a lot. So first, can we use NGS to detect viruses in environmental samples from live bird markets? Okay. I think for this crowd, we all understand that's a yes, but for a lot of people I talk to that say, oh, well, really, can you use, air to look for metagenomes? Yeah. And so if we look at a single bird sample, you can see this is an influenza-infected bird. We're getting 98% of the reads in a pool of 5 birds, right? So 98% of the reads that we're getting out of that bird, if we're just looking at viruses, are influenza. And then you can see some other, avian-associated viruses, which makes sense, right? Because it's a pool of 5 birds. They're going to have multiple viruses in them and things like that. If we look at a single air sample, what you end up seeing is that same influenza virus, which is about 15% of the sample, and you get all of these other viruses along with it. We see a lot of rotaviruses. I don't know if you guys see a lot of rotaviruses in your reads, but we see a lot of different rotavirus species, but a lot of those other avian viruses, but also things like bat, human, swine, cattle. You can imagine what's going through this market any day, any time as well. So, but how does it compare to gold standard PCR? So also a lot of people ask, but is detecting something in metagenomics really comparable to us doing our gold standard PCRs? So if we look in things like poultry swabs or wash water or even air, again, avian influenza is very, very prevalent in Cambodia. It's detected in 20 to 50% of birds at any given time. So here I'm just showing you basic flu A detection. It's not necessarily H5. But if we look with our metagenomics, that increases our detection by about 20%. Okay. And people go, why is that? Well, why are you actually seeing an increase? Does it mean that NGS is more sensitive, quote unquote, than PCR? And no, it does not mean it's more sensitive. It just means that we're able to get more of the virus in what we're looking at, right? So if we look at this is just a spiking experiment where, we spike different CT levels. This green line right here of 40.5, that's negative on PCR, okay? Clinically, that would be negative. But you can see how much of that genome we're actually getting at almost 100x coverage. So does that really mean that sample's negative? It may mean that it's not infectious, yeah? But if we're just worried about detection, it's still there, right? Okay. So this really requires a paradigm shift for how you use this data, right? If you're looking clinically, of course, we're going to need to worry about if things are infectious, if things are, transmissible. But if we're looking at basic metagenomics and understanding high-risk areas, if I went into a market and I got this, this coverage for, say, Ebola, yeah, but I got a negative PCR for Ebola, I'm going to worry that there's Ebola somewhere in that market. Okay, so, I think it's a question of how we use the data overall. So we don't really know what the real cutoff is. We all need to work on this together to make guidelines and things like that. But of course, it'll matter, as you've heard Aaron talk about things like, it matters what you're looking for and why. Okay, but from a policy standpoint, we need to really think about this, because people will start using this method kind of willy-nilly, and we won't know what's going on. In terms of what we see, this is just to be clear, this is just twist amplification on these samples run through CZID. Okay, we've done this a number of different ways with CISPA and with shotgun metagenomics, with a lot of different pipelines. But just for the sake of this talk, to show you patterns, I'm just showing you CZID data with twist. Okay, so take that with a grain of salt from everything that we've talked about this week. I think what I want to show you here, and what I think is evidently clear, is that if you look in drinking water and cloacal and tracheal swabs and carcass and wash water, you see a good amount of viruses, things like that. Again, these are viral families on the side, or genre on the side. But look at the air. Okay, we're seeing a lot of things in the air. And when we do statistics on this, and I didn't put this in, when we do statistics comparing the birds themselves, the swabs, and the air, we, down to the contig level, can detect everything we detect in the birds in the air. Okay, so basically every single contig that we have in a bird, we detect in the air at the same time, plus a lot of other things. So if we're detecting 70 viruses in a bird, or bird samples overall, we'll have 370 viruses in an air sample afterwards. Okay, so we find it's statistically very nice, and it does recapitulate what's going on in these markets. We're working on other things now. Obviously, this is from abattoirs. You can see this is a bit more messy, but things like carcass wash water, rope saliva, snout swabs, again, not so many viral genre, maybe a little bit down here, but again, a lot of diversity in that air. Again, it reflects what we're detecting in the swabs, plus more. And then things like bats. Again, if we're looking at air samples in caves and things like that, we're detecting a lot of different kinds of viruses that we may only be detecting in certain bats or things like that, but we'll detect them in a lot more. And then you see very nice patterns like this chunk right here that's really prevalent in the bats, but not really prevalent in the air. Those are actually papilloma viruses, which are sexually transmitted, right? So you'd expect that they're not really in the air. So I'm starting to feel like this is an okay way of looking at these things, and the more and more we do it, and the more people that are doing it, I think we'll see that it's quite nice of a method of using things. So the next thing is NGS actually comparable to QRT-PCR and ES? So can we actually look at this statistically? So if you look at things like reads per million or total reads, this is for avian influenza. Again, you see it's pretty messy, right? But the correlation does actually statistically work. So if you're looking at total avian influenza reads or reads per million over the entire genome, you actually do get statistical correlations, and this is a lot of work that's been done by Jay. But what we also get asked is, how does that actually correlate to the actual PCR? So if we look down at the actual gene we're looking at for PCR, again, we retain that statistical correlation. But then if we look down to even the tiniest base pair for that gene, you still see that statistically those are correlated. So I think we're continuing to do the statistics here on not only M but also H5, other genes, and we're trying to pull apart the different environmental samples and how that works. But it's quite encouraging that it seems like you can use certain statistics from your metagenomics to actually tell you how much viral load and things like that is there from what you've done, as long as you control appropriately and things like that. Finally, are environmental samples actually able to effectively replicate what is detected in poultry in the market? And as I told you, we can actually effectively replicate things down to the contig level, okay? But you're probably saying, okay, why do we care, right? Why you care is that you can pull out full genomes from these air samples, and we do it quite often, and we can use them to compare to duck and chicken and other samples. Here's a very nice example right here where you're seeing a duck this is the 4th of April 2023, same day and same time. We pulled the same virus from the duck and the environmental sample, and they're exactly the same genomically, okay? Exactly the same. And you see several other places where we're seeing in the air, also in the wash water in the cages. We see also up here, we have different duck and environmental samples that are all matching. But what's important about this too is that we are detecting things in the air that we're not detecting in the ducks and the birds and the chickens. So if you imagine there's 10,000, tens of thousands of birds going through this market every single day, and my team goes in and samples 40 of them, okay? Can imagine and we're still detecting 20 to 40% positive, but you can imagine how much we're missing. So what we're actually seeing in the air, if we just look at something like H5, is that there are days we can actually detect multiple subtypes of H5 going through the same market, okay? And so if you think back to the reassortment of H5 and human infections in Cambodia, this is important. So we were actually able to pull apart different subtypes of avian influenza in the air going through the market that we completely missed in the birds that day. Okay, that's a really important piece of information, especially for flu that reassorts a lot. So, in my opinion, this is one of the most important things that we're seeing. So, can we actually effectively replicate what is detected? Yes, but there are caveats, and I speak really nicely of this. But again, there's limitations to everything, and I'm happy to talk about those later. But yes, we may need to take multiple environmental sample types. So take a water, a swab, an air to compare, but that's really easy compared to sampling 40 birds and everything else. The last thing I really wanted to touch on, and we've been touching on this the other day, so I decided to throw this in last night, was really that, and this is real, by the way, this is real from the market. You can see his tire is way off the ground. But how do we make this heavy burden of data that we're getting from metagenomics, especially for things that like we do in Cambodia, which is not necessarily discovery, it's more how do we use it for action? How do we make it actionable, reportable? And that's really, really difficult. And so we've been thinking about this for a long time. We've held workshops on this. We've worked with the Cambodian government. We're working with the South African government, actually next week in Durban, with the municipality of Durban on their wastewater supplies. But, really, of course, now we still have to focus on priority diseases. People want to know about COVID. They want to know about polio. They want to know about avian influenza. So we still have to focus on those priority diseases and what the priority is of the country, and that will change over time. So when you're doing your metagenomics and you find something, it may not be a priority in your country, but it may be a priority somewhere else as well. We also need to continue to validate. A lot of policymakers and things like that, they say, well, is this validated? Did you report it to WHO? Did you report it to WOA? And until we actually get validation of a lot of these detection techniques, it's quite hard to use it for policy in action or even clinical decision making, right? So I think we all need to continue to validate what we do when I know we talked earlier about running PCRs and things like that to check or isolate, but every time we can do that to show that these technique works, that adds to the evidence that it works, okay? So I know it's extra burden, and I know people don't want to do it. I know they want to get papers out, but if you have a chance to run the PCR, run the isolation, at least after you get your paper out or something, yeah? So we have the data. We have to partner with a lot of stakeholders. So, we have to tailor the analyses that we do depending on the context of who we're talking about. Talking to a Ministry of Health is much more different than talking to a Ministry of Ag or to anyone like that. So we have to really understand what each individual stakeholder needs so that we can actually use the data for good. We can't just make a dashboard and say, read my dashboard, right? It doesn't work that way. We also have to provide data visualizations. And so we've held a workshop in Cambodia where we literally said, do you guys like heat maps? Do you like line bar graphs? Do you like bar graphs? What colors do you like? Do you like purple and green together? Do you like red? What does red mean to you? Does it mean warning? Because it's different in different cultures. And I really encourage you to go talk to your policymakers and say, what kind of graphs do you like? Because I've seen reports where people have these beautiful graphs, but no one understands them because, you know, that they're made, and maybe Spiros will talk about this later in the week, but, you know, really focus on when you're generating reports for action that your policymakers want. I also believe that we need to automate these identifications and set alerts, especially when things go above thresholds. And then for me, you know, I think the most important is using the data differently, shifting the paradigm that this is not just detection. So can we actually use viral diversity measurements or reads per million or other things to not only understand the risk of an area or what's coming out of a hospital or things like that, but how can we also use it to understand how well our interventions are working? Okay, so if we clean up the market, can we actually measure the diversity go down, right? Or if you put 6 people in a triage room, and you see the diversity go up, and so you can know clinical decisions saying, let's not put 6 people in a room, right? And I think using those diversity statistics and other things are really, really useful tools. But again, we have to change the way we think about it. So with that, I'll stop. I think I'm not too bad on time. I work with a lot of people. We have a lot of different partners, and I'm contactable at many, many different places. So thank you very much.
Fantastic talk, Eric. Do we have any questions from the audience?
Yeah, really exciting talk. So I'm a medical doctor who is kind of keen to switch more into One Health area. So we're very interested in looking into wild birds in the area. And I was wondering what your experience is with air sampling that is not like, because those markets feel like an enclosed environment. So would air sampling like really in an outdoor space also work, you think?
There have been people that have been trying it. I know they've tried it in Australia. People like Paul Horwood, Michelle Wiley. They are trying to like make these tripods that they put in the middle of colonies. You really have to get in the middle of the roosting site. I think it would probably work as long as enough is being stirred up and you let it run for long enough. What I've seen work really well, especially if you're looking for avian influenza in wild birds, is water sampling. So a lot of times, obviously, the natural reservoir is water birds, right? So people are doing really, really nice studies in Italy and things like that, where they're using these basically passive torpedoes for water sampling and things like that, that they're picking up avian influenza in the Bay of Venice, and then they start seeing outbreaks in the middle of Italy about a week later. So you can actually predict when the virus will actually get into the country by measuring sort of those edge areas as well. So there's a lot of ways to do it, and it's definitely worth trying.
And what is with sampling in these farms? Like, are you well received in all of these farms to search for these?
So we're always with the government, right? So we always go out with government partners. And most of the time, we're not handling the animals ourselves. It's the national vets or the local vets that are handling animals, and we're taking environmental samples at the same time. Generally well received. Jay probably has some more stories than me if you want to talk to him after the talk. The only times that we get some kickback is we just like did an intervention study in this market, and I know the sellers were kind of annoyed that we were there all day for like 7 days, you know, for 4 hours, right? So when you disturb people, that's the problem. But when they're used to you coming in and doing longitudinal work, a couple hours here and there, it doesn't seem to cause a problem. Plus, you know, most people get a bit of a incentive for that.
Thanks for the talk. I'm assuming the air capture devices are not single use. How on earth control for contamination and like the, you know, it must be a nightmare to compared to anything else.
Meaning what? What are you considering contaminating your air sample with?
Everything in the air, I guess. But I mean, if
we
you
want to know what's in the air, right?
but if
So-
you're using it once and then you
Right. So a lot of these are single-use filters, right? And I didn't show pictures of it here, but like those big filter air samplers, they basically have a syringe with like a sponge in it, and you screw it in and you push it in so it's sterile. yeah? And then you lock it up, and the only thing going through that sponge is the air that it's pulling through the machine. The smaller ones that you saw Jay working with, those have a cup basically that goes on the bottom. It has like a gel matrix in the bottom. And so it's only going into that cup of what we need, right? And we have seen weird stuff in our samples, whether that be from our lab, you know, of course you get lab contaminants and things like that, or, and we can debate this, you know, things like mosquitoes and stuff will fly into your air sampler. We've seen it happen. So if you may pick up a weird virus in the air and be like, this shouldn't be airborne, but you may have a mosquito or something in there, right? So, and they get very dirty, and there's a lot of other stuff in there, but we always also try to run an air sampler way far away from the risky area to use as a sort of control, which was what was interesting about running in the food sales in the market there, because we were still detecting avian influenza even that far away from the market. So it can actually tell you some really interesting things.
Great talk. Thank you so much. I just have a question with regards to in Cambodia. What are the policies like in terms of the human testing labs interacting or tapping into the animal testing labs? Because I know in South Africa we struggle a lot. We are trying to get a Section 20 currently in my lab just to get the One Health situation working.
So legally, under Cambodian law, I am the only lab in the country that's allowed to handle animal and human samples in the same place.
What did you do? Give us.
I think we kind of got grandfathered into that, honestly. But we have facilities. So we have a P3 facility, but we also have P2 plus facilities for both animal samples and human samples that are separated. Okay. Under the law, the animal labs are supposed to test animal samples, and the human labs are supposed to test human samples. You can imagine how awkward that gets when you're looking at zoonotic diseases, yeah, things that come from animals, or when you're looking at environmental samples. And the environmental, you know, labs don't really have the facilities to do testing like this. A lot of it's food and water safety and things like that. So this whole One Health ecosystem really, again, has to change the paradigm of what labs are allowed to handle what labs are in charge of what. And that might not be happy to some people that are in this room, but you know, PCR is PCR. Yeah.
You know what my question is going to be, but obviously
Do I?
I'd be keen to know if you've done air sampling in hospital wards and whether you can then link the air samples to the patient samples. What you get.
We have not. I wish I had, because we have all these H5 cases in wards. We're working on it for next year because a lot of the wards are kind of open, especially the emergency wards. They're packing 10, 20 people in beds into these rooms, and then their families in there with them. So being able to correlate that with individual cases, with wastewater coming out of the hospitals, will be very, very interesting, and it's something that we have planned as well. Everyone ready for wine?
I have a first.
Aaron's keeping us.
Yeah, sorry. So are you using any kind of normalization across your sequencing libraries, like something like a spike in? And tied to that question, do you see this being a method that could be rolled out for like routine surveillance and maybe combined with some kind of AI bioinformatics tool to sort of be an early detection for re-emerging but also novel pathogens? I mean, if you see something suddenly popping up everywhere, it could be worth investigating.
We don't use spike-ins. It's something we're considering as well, at least talking to other collaborators, how to normalize these things. We were using equal number of reads across samples to kind of normalize, you know, what we could. But I think in the future, if we're going to do this on a routine basis, of course, having an internal control, just like you would have for PCR, where you're looking at, you know, a human gene or anything like that to know that it's actually worked, I think is critical for this kind of decision making. Ideally, I would love, I mean, we wrote a paper at one point called Smart Markets, right? where, and again, using these triggers to sample air. And then I would love these automated platforms that would just take that sample and do the analysis and do AI and tell us that some big bright light comes on in a room somewhere that says, go to this market and fix it. That's where we're going to with dashboards, right? So we're using dashboards to really make this metagenomic data accessible to decision makers, and really kind of working that out. But right now it's very slow, because I think we all know it takes a while to get this data back to the lab, or the samples back to the lab, get it all done and through. So anything we can do to speed it up, I'm happy to look at. But AI, I have a little bit of worry about making up, the next pandemic or something, or inserting, a base pair somewhere.
Have you also seen any kind of evidence that your reporting is sort of leading to meaningful change within the government? Do you think that they're sort of listening to you? And I mean, often we say things to governments over and over and over again that, something is concerning, and until it's kind of already turned into an issue, they kind of don't want to listen. Do you think that you'll see change in Cambodia in relation to the exposure of people to viruses?
Exposure is an interesting.
Well, like changes to the wet market.
I mean, I think that's what we're trying to prove right now. A lot of countries, because we don't just work in Cambodia. We're employing these air samplers now in Vietnam, Indonesia, Laos, Nepal, Tanzania, Senegal. I think they're getting some. Guinea. There's talk about using some of our stuff. Well, there was talk about using some of our techniques for Ebola in Rwanda when there was an outbreak. I think everyone's really interested in using it, but the question is, how are they going to use it now? I know the government of Cambodia is going to start integrating a bit more of this environmental surveillance in what they do outside of us, which is great. I think that's very interesting. But again, it's going to take some time to integrate that to national policy. So I welcome all of you to give us your thoughts on how that could be done better.
More questions? Okay, everybody's ready for a while. So thank you, Erik.